So this is summary. You can see insulin, glycogen synthesis in fed condition. So insulin will be there, and then in liver, glucokinase, hexokinase, and all kinases require magnesium. Wherever ATP comes, magnesium will follow. Okay, ATP is followed by magnesium always. So remember, that is why magnesium is always there. You can see this is the glycogenolysis. Though it is shown here down, but you have to remember it is uphill process. So you can see the synthesis energy requirement. That is how you can connect energy requirement. Now this is downhill reaction, but see, in downhill reaction, you did not see actually utilization of energy. A very unique mechanism you can see from glycogen. You have seen in muscle glucose 1-phosphate is released. Suppose from glycogen there would be production of glucose. So what would happen? So this glucose would require one more ATP to convert into glucose 6-phosphate. So in this process, glycogen phosphorylase is a very unique type of enzyme that actually conserving energy during glycogen breakdown, especially for muscle, by converting this. So glycogen actually breakdown is very energy efficient process. Okay, this is efficiency of our body that produces glucose 1-phosphate. Otherwise, glucose, if there were production of glucose, maybe because of hydrolysis, because in our intestine hydrolysis produce, because this glycogen will be converted into glucose in our intestine by hydrolysis. But here in our cell, the enzyme is very different, that actually add phosphate, through phosphate that add. This is the advantage of phosphorylysis versus hydrolysis. Enzyme in intestine converts into glucose. Hydrolysis versus phosphorylysis. Advantage of hydrolysis versus phosphorylysis. Whatever concept, whatever comparison, contrast I do, that becomes conceptual things always become important for USMLE. Okay, and you will not find these conceptual things anywhere. Whatsoever video you watch, you will not see there. So that is why I try to grasp those concepts. So compare this phosphorylysis with hydrolysis. Phosphorylysis is energy efficient process in comparison to hydrolysis. So let's talk about this quiz. In response to glucagon and epinephrine, cells undergo a series of changes in signal transduction inducing molecule that phosphorylates and activates glycogen. Which of the following enzyme is responsible for covalent modification of glycogen phosphorylase? Yes. Read carefully. So there are two choices. You have phosphorylase kinase also. So when there is two choice, you have to choose most appropriate. Yes, A is not wrong. Okay, A is not wrong. Protein kinase A, but indirectly. Directly, this is phosphorylase kinase. Okay, so you can see here this phosphorylase kinase. This actually activates phosphorylase kinase by phosphorylation. So this indirectly, this is, you are right, but most appropriate will be this. So did you agree with me? Do you agree with me?
Yes, sir.
Okay. Now, which of the following is not the direct or indirect? Okay, activator of glycogen phosphorylase, which is inhibitor. D is inhibitor. Okay, D is inhibitor. This is also activator, this is also activator, this is also activator. Which of the following statement is false regarding glycogenolysis? Glycogenesis. Following statement is false. Glycogen synthesis is activated in phosphorylated state. Synthesis is inhibited by glucagon and epinephrine action. Protein phosphatase removes phosphate group and activates enzyme. And then insulin promotes glycogen synthesis in liver and skeletal muscle. So, which of the following statements is false? Glycogen synthase is activated in the phosphorylated state. Yes, it is not activated in phosphorylation. And this protein phosphatase, by removing many enzymes, are activated. So this statement is partly true. Not always true, but partly true. Glycogen synthase enzyme is inhibited by glucagon. That is true. Insulin promotes glycogen synthesis. That is also true. So this statement is partly true. Okay, mostly true, and false in case of phosphorylation. But this is totally false. Now, glucagon and epinephrine stimulates glycogen breakdown to glucose 6-phosphate directly by binding. So you can see this. Glucagon and epinephrine, these are hormones. Breakdown of glucose to glucose 6-phosphate. How? It is always indirectly. I've already told you. Now, glycogen synthase is activated by phosphorylation. Does phosphorylation activates glycogen synthase? Dephosphorylation. Okay. B is the answer is. And then compounds responsible for coordinated regulation of glucose and glycogen metabolism are this. You mean this?
Yes.
What about this?
TCA.
And this also. See the compounds. So I want to give you one concept here, okay? The concept I told, you have to remember, glucose converts into glucose 6-phosphate, okay? So here from glucose 6-phosphate, glycogen will be formed, okay? Glycogen. And then glucose 6-phosphate will convert into fructose 1,6-bisphosphate. Okay. And this will also stimulate this reaction. I'll change the color so that you will understand. This will stimulate, okay? Now, I'll tell you, if you see the glycolysis will progress, okay? And pyruvate will form. And if glycolysis is aerobic, it will convert CO2. From this, NADH will be produced, ATP will be produced, and then, you know, from here also NADH will be produced. So here is the enzyme that is called fructose. This is enzyme is called. This is the very important rate limiting enzyme, the most important rate limiting pacemaker step, PFK1. So now you see this, when glucose is high, so there will be very high amount of fructose 2,6-bisphosphate. It will activate. More glucose becomes high, more this will be there. But the inhibitor, these are the inhibitor that will actually inhibit this. These are the ATP and these are the inhibitor that will inhibit this. And because of inhibition of this, what will happen? There will be actually production of more glycogen. Inhibition of this, and there will be also inhibition of pyruvate kinase by this ATP. And there will also be inhibition of pyruvate dehydrogenase by this ATP acetyl coenzyme A. So this inhibitor will block this reaction. As a result, this will be accumulated, become high. And then the pathway will be sluggish, so this pathway will be stimulated. So actually feedback inhibitors are of glycolysis actually push glucose 6-phosphate to glycogen. So these are the actually feedback inhibitors actually push this glycogen to this, and also this feedback inhibitor are the one that will push this glucose for fat synthesis. So thanks to this feedback inhibitors, this feedback inhibitor push things for fatty acid synthesis and glycogen synthesis. Glucagon is released from pancreas. Okay, insulin and glucagon both released from pancreas. Glucose 6-phosphate is present only in phosphatase,
Liver.